The overall goal of this procedure is to measure the amount of free radicals in mainstream tobacco smoke and to assess the scavenging capacity of several antioxidants. This is accomplished by first preparing filters containing several different scavenger compounds, as well as controls without scavenger. Filter preparation is followed by smoking simulation, with the smoke of research cigarettes passing through a spin-trapping solution. Electron spin resonance, abbreviated ESR, is then used to record spectra of trapped radicals as well as of reference samples. Next, obtained ESR spectra are subjected to routine analysis in order to separate the signal of the trapped gas phase radicals from the background and to quantify the radical concentration using a reference sample. Ultimately, results show quantitative information on the content of free radicals in mainstream cigarette smoke and estimates of scavenging capacity of natural antioxidants through spin-trapping ESR spectroscopy.
The main advantage of this technique over the existing methods of tobacco smoke analysis, like gas chromatography, is that ESR is specifically sensitive to free radicals. The implications of this technique can help in reducing hazardous effects of tobacco smoke because free radicals is a major group of carcinogens.
This method can provide insight into studying free radical in the gas-fed smoke, and it can also be applied to other systems such as industrial smoke, exhaust, and smacks.
The National Biomedical Research Center for Electron Spin Resonance Technology, also known as ACERT, is an outgrowth of our extensive experience.